Hi, hello NEET and EAP set aspirants. This is your Prasanna ma'am from NEET World. Now we are going to discuss about the chapter Anatomy of Flowering Plants. To analyze and understand this chapter in a better way and to score complete marks from this chapter, you have to learn this chapter in 3 fragments. First is tissues, second is tissue systems. After learning tissue systems, you should learn the proper comparisons between dicot, monocot, root, stem and leaf. Clear? And thirdly, you have to focus on the secondary growth. Of course, secondary growth is not there for NEET, but then for EAP set students this is important. Okay, definitely 1 to 2 questions will be covered from tissues. 2 questions will definitely come from tissue systems, 2 to 3. Okay, tissue systems and comparisons between dicot, monocot, root, stem and leaf. And EAP set to pakka there will be question from secondary growth. So this is the planner on how to deal with this chapter. So now within 20 to 30 minutes let us have a quick discussion on tissues so that we can solve all the objectives. Firstly, what is anatomy? Anatomy is nothing but the study of internal structure of plants. When you transversect root, stem, leaf and observe the internal structure of that is called as anatomy, study of internal structure of plants. Now, let us know the proper definition of tissue, and this is important in the objective perspective also. Tissue is nothing but those cells, group of cells which has common origin and perform common function. Group of cells which have common origin and perform common function, they can be structurally similar or dissimilar. They can be structurally similar or dissimilar. Clear? Now, plant tissues are classified based on the origin, position. They are classified. Firstly, they are classified as meristematic tissues and permanent tissues based on only one term, that is whether they can divide or not. Okay, meristematic tissues whose only activity is cell division. Meristematic tissues only activity is what na? Cell division. Permanent tissues cannot divide. They have, each permanent tissue have their own unique role of function. Unique functional responsibility is there for each and every permanent tissue. So those cells which are continuously dividing are placed under meristematic tissues. Again, based on the time of origin, they are classified into primary meristems and secondary meristems. Primary meristems are further classified based on the position. Based on the position, if they are present at the tip of shoot and tip of root, they are apical meristems. For example, root apical meristem and shoot apical meristem. Okay, and intercalary meristems are present in between 2 nodal regions. Intercalary meristems are present in between 2 nodal regions. Secondary or lateral meristems are classified if they are present in between xylem and phloem or in the stellar region, then they are called as vascular bundles. Those vascular cambium which is present in between xylem and phloem is called as intravesicular cambium, and the vascular cambium which is present in between 2 vascular bundles is called as intervesicular cambium. The vesicular cambium or vascular cambium which is present in between 2 vascular bundles connecting to intravesicular cambium is called as intervesicular cambium. Cork cambium is present in the cortex region. The only role of secondary or lateral meristems is to increase the girth of the plant, or the size of the plant. It is mostly seen in the gymnosperms and woody plants. Okay, so yes, we have meristematic tissues based on the time of origin. They are classified into primary meristems, secondary meristems. Primary meristems again based on the position, they are further classified into apical meristem and intercalary meristem. Apical meristems are of two types, root apical meristem, shoot apical meristem. Secondary or lateral meristems only function is to increase the girth or the size of the plant. If it is present in the stellar region in between vascular bundles, it is vascular cambium. If it is in between xylem and phloem, it is intravesicular cambium. If it is in between two vascular bundles, it is intervesicular cambium. If the secondary or lateral meristem, it is present in the cortex region, then it is cork cambium. Now, coming to permanent tissues, if the tissue is formed by similar type of cells, it is formed by similar type of cells, which are structurally similar. If the simple permanent tissue means the tissue is formed by structurally similar cells. Those tissues are of three types: parenchyma, collenchyma and Sclerenchyma. Clear ma? Complex permanent tissues are formed by structurally dissimilar cells. It is of two types again, xylem and phloem. Xylem has four tissues in it, which includes tracheids, this includes tracheids, vessels, xylem fibers and xylem parenchyma. Phloem includes four different tissues which are different in their structure as well. Those are sieve tubes, companion cells, phloem fibers and phloem parenchyma. Clear? This is the classification of plant tissues. You have to remember it for sure. Okay. Now let us exercise or understand each and every type of tissue in a more detailed way. If you look into the meristematic tissue's main function is to grow and divide, and these are immature cells. Meristematic cells are what ma? These are immature cells. They are of different shapes like isodiametric, round, oval and polygonal, and there is no intercellular space between two cells. Between two cells there is no intercellular space. The walls of these cells are thin, elastic and they are made up of cellulose. Nucleus is conspicuous and cytoplasm is dense. As they are immature cells, nucleus is conspicuous and cytoplasm is dense. Vacuoles are completely absent or they are small, and most important thing about meristematic tissue is as they are dividing continuously, their respiration rate is very high. The respiration rate of meristematic tissue is very high. So, these are the important features of meristematic tissue. If you want, you can take a screenshot of it. Most important points ma. Now, this is an NCERT diagram. Most important, you look into it. See, root apical meristem and shoot apical meristem. Root apical meristem and shoot apical meristem. Okay. So, here root apical meristem is closer to the root apex. Root apical meristem is closer to the root apex. It is in the subterminal region. It is in what region ma? Subterminal region. What is at the terminal region? Root cap, and root cap is from the initials of the root cap. Here are the initials of the root cap, and root cap is at the terminal position. Just above and below, closer to the root apical meristem, you find the initials of the central cylinder and cortex. Closer to the root apical meristem, you find what ma? Initials of central cylinder and cortex. Over it you have protoderm which later matures to epidermis or epiblema. Okay, and here at the peripheral sites you have cortex, and in the center you have central cylinder which has conducting tissues like xylem and phloem. This is all about root apical meristem. Coming to shoot apical meristem. Shoot apical meristem is from distant most region from the stem axis. Shoot apical meristem is at distant most region distant region from stem axis. It is at the distant region from the stem axis. Okay. So here above it you have leaf primordia, and at the axial region, the angle between stem and the main branch, the axial region, auxiliary bud is formed. Okay, at the axial region what is formed? Auxiliary bud is formed. This auxiliary bud may develop into branches or flower or a pedicel of a flower. Okay, this auxiliary bud may develop into leaf, branches or a flower. Okay, next, and here you can find the differentiating vascular tissue. I hope both the diagrams of root apical and shoot apical meristems are clear. Now we will look into the types of meristematic tissues. Of course, we have discussed root apical meristem and shoot apical meristem, which will increase the length of shoot and root. Intercalary meristem, which is present in between two nodes of the stem, it will also perform the same function, that is increase in the length of the stem. But then one more unique function which is applicable to grasses. Whenever grazing herbivores eat these grasses, the regeneration of the growth is happening. What is happening, beta? Regeneration of growth happens because of the intercalary meristems which are present in between the nodal regions. These intercalary meristems will ensure the growth of the stem again, even though they are destructed by grazing herbivores. Okay, this is the unique function with respect to intercalary meristems. Now, if we look into the secondary meristem, as we have discussed, secondary meristem's main function is to increase the length of the plant. As they are appearing in the later part of plant's life, it is called as lateral meristem. As it is after the primary meristem, it is called as secondary meristem. So, to analyze the secondary meristems in a better way, if you take the basic structure of the plant, here you can see the stellar, just a second, here you can see the, just a minute, stellar region wherein you have xylem and phloem. I am taking a dicot stem where you have xylem towards the closer to the pith region and phloem towards the peripheral region. Okay. So, the cambium or the few cells in between xylem and phloem get meristematic again, and they form a cambium between xylem and phloem. This is the intravesicular cambium. The cambium which is formed between two vascular bundles connecting the intravesicular cambium is called as intervesicular cambium. And this is intravesicular cambium. And the cambium which is formed in the cortex region is called as cork cambium. It is called as what, ma? Cork cambium. Fine. This ensures the stellar secondary growth. This ensures non-stellar or cortical regions secondary growth. Clear? So, here you can see cork cambium, intervesicular cambium and intravesicular cambium. Clear, ma? This is about secondary meristem. Now, if you look into permanent tissues, what are permanent tissues? Those tissues which are devoid of cell division, and they have specific and they have specific functional role. Functional role. They are of two types. One is simple permanent tissues. Second is complex permanent tissues. Simple permanent tissues will have all the cells which are similar in structure. They are structurally similar. As such, we have three types of simple permanent tissues. Those are Parenchyma, Collenchyma and Sclerenchyma. And complex permanent tissues are the conducting tissues in the plant. Those are Xylem and Phloem. Okay, yes, xylem we have four different tissues which include tracheids, vessels, xylem fibers and xylem parenchyma. Phloem again has four different tissues which includes sieve tubes, companion cells, phloem fibers and phloem parenchyma. These are the tissues which we have to learn under the permanent tissues. Let us learn it one by one. So, yes, simple permanent tissues, of which the first tissue is parenchyma. Parenchyma is in almost all the plant body. Across 80 to 90% of the plant body is filled with the tissue parenchyma only. Okay, almost 80 to 90% of the plant body is filled with the tissue parenchyma. It is abundant permanent tissue. If there is a question in your exam, what is the abundant permanent tissue? Abundant permanent tissue in the plant body, then your answer should be parenchyma. Clear? These are, the cells are isodiametric, means all cells are similar in structure. They may be of different shapes. Okay, they are isodiametric and they may be in different shapes like spherical, oval, round, polygonal and elongated, but all the cells are of same structure. Okay, now their walls are thin and made up of cellulose. The cell wall of parenchyma are thin and made up of cellulose, and they can be packed or they can have small intercellular spaces, not like meristematic tissue where there is no intercellular space. Parenchymatous tissue can have little intercellular spaces, and each cell has, at the center, each cell has elongated vacuole, and at the peripheral region they have cytoplasm, Internally, each cell encloses large central vacuole and peripheral cytoplasm, and they contain a nucleus. Nucleus and cytoplasm are at the peripheral region. Centrally, they have an elongated vacuole. They are covered by cellulose, thin cell wall cellulose, and intercellular spaces may be not at all present, or may be thin intercellular spaces are possible. These are the structural features of parenchyma that you have to remember. Now, types of parenchyma. Based on the functions, we have different types of parenchyma. If at all parenchyma is having chloroplast in it, okay, parenchyma is having chloroplast in it and conducting the function of photosynthesis in leaves, then it is called as Sclerenchyma. Then it is called as what, ma? Sclerenchyma. Clear? And if it is in aquatic plants, parenchyma has large air sacs or air spaces in between, where you will call it as aerenchyma. The main function of aerenchyma is to promote buoyancy, okay, in aquatic plants. In what plants, ma? Aquatic plants. This is one more type of parenchyma. Last type is storage parenchyma. Mostly seen in some succulent plants like aloe vera. Okay, clear? So, these are different types of parenchyma. Now, Yeah, functions of parenchyma include they act as storage parenchyma and store the food material, and they can provide the turgidity to softer parts. They can provide the turgidity to softer parts and protection from the epidermis. They form the protection in the form of epidermis. As I already told you, most of the plant body is composed of the tissue parenchyma. Epidermis is also made up of parenchyma, providing the role or promoting the role of protection. Okay. Next, lateral conduction in the form of xylem and phloem. Xylem and phloem are also parenchymatous tissues only, like, and photosynthesis in the form of Sclerenchyma we have discussed just now, and they provide buoyancy as in Parenchyma. These three are most important functions you have to remember, and protection in the form of epidermis, lateral conduction. Okay, conduction between xylem and phloem of different vascular bundles. Lateral conduction is promoted by parenchyma. Clear? Now, Collenchyma. If you look into the structure, cells may be oval, spherical, polygonal, or often they contain chloroplasts. Okay, and it is found either as a homogeneous layer or in the form of patches. Okay, and the cells in the Collenchyma are much thickened in the corners, and the cell wall composition is very important. You have to remember it includes cellulose, hemicellulose and pectin. Okay, the cell wall composition of Collenchyma includes cellulose, hemicellulose and pectin. These cells assimilate food when they contain chloroplast, and intercellular spaces are again absent. Short intercellular spaces are present in parenchyma, and thin cell wall with only cellulose composition is present in parenchyma. Here if you see, they have thick cell wall composition which includes cellulose, hemicellulose and pectin. Let us look into the functions of Collenchyma. The functions of Collenchyma include provides mechanical support. As they have tough cell wall, they can provide the support, mechanical support to the young stem, petiole and leaf. Important function. Next, it provides flexibility to the petiole and allows their bending of leaves. It provides flexibility to the petiole and allows the bending of leaves. It prevents the tearing of leaves. Okay, it prevents the tearing of leaves as the tissue is more strong and tough. It helps in growth and elongation of organs also. These are the important functions with respect to Collenchyma. Next, Sclerenchyma, if you look into, this is the dead tissue. Okay, Sclerenchyma is a dead tissue. It does not have a cytoplasm or conspicuous nucleus. It is a dead tissue. The tissue occurs where the sclerenchymatous tissues you can see in hypodermis, vascular region, little areas of cortical region and stem leaves. Okay, stems and leaves. Structure includes Sclerenchyma consists of long and narrow cells. It consists of long and narrow cells, thick and lignified cell wall. It has thick and lignified cell wall. If you look into the cell wall of parenchyma, it is only cellulose. Cell wall of Collenchyma, cellulose, hemicellulose, pectin. Cell wall of Sclerenchyma is lignified. It has few to numerous pits. Okay, it has few to numerous pits. Now, they are usually dead without protoplast. That is no cytoplasm and nucleus. It is of two types, fibers and sclerites. Fibers has narrow lumen, sclerites has very much narrow lumen. Okay, it has narrow lumen and the cell of fiber is elongated. You will see the fiber type of tissues in xylem fibers and phloem fibers. Sclerites are also seen in pulp of fruits and fruit walls of nuts. These areas sclerites are present. Okay. Now, if you see the differences between fibers and just a second, fibers and sclerites. Fibers include fibers. If you look into the shape of fibers, they are elongated, thick walled and pointed. This you will see in xylem fibers and phloem fibers. And coming to the shape of scleroids, they are spherical, oval and cylindrical. Cell condition, both are dead cells because they are sclerenchymatous. Lumen is narrow here, here you have very narrow lumen, and their occurrence in various parts of the plant, they are scattered in the specific parts. Function, they provide mechanical support, fibers will provide mechanical support, they provide hardness and mechanical support. What are the examples of fibers? Like commercial fibers like jute, flax and hemp. Scleroids are present, most important many at times questions are asked, where are scleroids present? They are present in the fruit walls of nut, pulp of guava, pear and sapota, and they are found in the seed coat of legumes. They are also found in tea leaves. Okay, these are the major differences, make a note of it. Next, coming to the functions. Sclerenchyma provides mechanical support to the organs, and scleroids provide strength to the seed coverings. Scleroids will provide strength to the seed coverings. Next, scleroid form the stony endocarp. Scleroids will form stony endocarp of many fruits and stone fruits, for example almond. Okay, scleroids will form the stony endocarp in many fruits. Coming to complex permanent tissues, this is the vascular bundle and xylem and phloem. Okay, and now we are going to exercise all the 4 different types of tissues which are present in xylem and phloem. So yes, if we look into xylem, the 4 different tissues, first it is tracheids and vessels. Most important thing you have to remember is the structure of each xylem tissue, its function. Most important thing is in what type of plants that tissue is present, that you have to remember. Tracheids. Tracheids are elongated tube-like cells. They are elongated tube-like cells. They have lignified cell walls, wide lumen and tapering ends. They have tapering ends. They are tube-like cells, lignified, they have wide lumen and tapering ends. These are dead and without protoplasm. They don't have any protoplasm. Thick walls with variable inner wall thickenings. Okay, they have variable inner wall thickenings, and they have pits. Okay, pits are present in tracheids. They are present in all the vascular plants including gymnosperms. So tracheids are present in pteridophytes, gymnosperms, angiosperms. Clear mass? This is all about tracheids. Coming to vessels, they are long and cylindrical. Okay, these are tube-like, but vessels are cylindrical tube-like structures. They are made up of many vessel members. Okay, each with lignified walls and central cavity. Each has lignified cell wall and they have central cavity. Vessel cells are also devoid of protoplasm. Vessel cells are also devoid of protoplasm. Vessel members are interconnected through perforation plates. These are perforation plates. Okay, these are perforation plates which connect to vessels, and vessels will perform more effective water conduction than tracheids. This is because of the presence of perforation plates. The presence of vessels is a characteristic feature of angiosperms only. In gymnosperms and pteridophytes, vessels are absent. It is characteristic features of only vessels are characteristic feature of only angiosperms. In gymnosperms it is absent. Now coming to xylem fibers. They are highly thickened cell walls. Just now we have learnt about fibers, right? They have highly thickened cell walls. They are dead cells with thick walls and obliterated lumen, means narrow lumen. Okay, they can be septate or aseptate, means divided or not divided. They provide mechanical support and they are present in all the vascular plants. They are present in all the vascular plants. Xylem fibers are present in all the vascular plants. Tracheids are also present in all the vascular plants, which means pteridophytes, gymnosperms and angiosperms. Okay. Xylem parenchyma, if you look into, it is a living component of a xylem. It has thin walled living cells. The walls are made up of cellulose. Xylem parenchyma will store starch, fats and tannins. We have ray parenchyma, which aids in the radial conduction of water. Storage of food and radial conduction of water is present, and they are also present in all the vascular plants. So, the only exception is for vessels. Rest all the tissues of xylem are present in all the vascular plants. So, what you have to remember with respect to xylem parenchyma, this is the only living component of xylem, rest all are the dead tissues, and they have thin cell wall, that is cellulose. They store starch, fats and tannins. The ray parenchyma will help in the radial conduction of water, and they are present in all the vascular plants. Now, if we look into the types of xylem, the primary xylem as it is forming, types of xylem, okay, most importantly primary xylem. Okay, we have two types of primary xylem. It is protoxylem and metaxylem. Protoxylem in roots is at the periphery, metaxylem is at the center, which is close to the pith region, So, protoxylem, which is the primitive xylem, okay, or first formed xylem, later it matures to the metaxylem. So, protoxylem if it is to the periphery, metaxylem to the center, it is called as exarch form of xylem, which is observed in the roots. Okay, it is observed in the roots. Endarch form of xylem, where protoxylem is closer to the pith and metaxylem is towards the peripheral region. Protoxylem is towards the pith and metaxylem is towards the periphery. This is endarch xylem. It is seen in the stems. Okay, it is seen in the stem. This is about the types of primary xylem. You have two types, protoxylem, which is the primitive xylem, and metaxylem, which later matures to metaxylem, right. If protoxylem is towards the periphery and metaxylem is to the center, it is exarch condition seen in roots. If protoxylem is towards the pith, metaxylem is towards the periphery, it is endarch condition seen in stem. Clear? Now, coming to the last type of complex tissue, which is phloem. Okay, in phloem, if you see, it is a complex permanent tissue composed of sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Of course, in gymnosperms, sieve tubes and companion cells are absent. Sieve tubes are present in the form of sieve cells, companion cells in the form of albuminous cells only in gymnosperms. In xylem, which tissue is absent in gymnosperm? Vessels. Remember it, okay. Coming to sieve tube elements, these are dead, okay. Sieve tubes are also dead tissues. They are long tube-like structures arranged longitudinally. This is your NCERT diagram. These are arranged longitudinally, and the end walls are perforated with sieve plates. Their end walls are perforated with sieve plates in which they have sieve pore also. Mature cells lack nucleus but contain peripheral cytoplasm and large vacuole. So, sieve tube cells will contain peripheral cytoplasm and large vacuole, but they are enucleated, they do not have any nucleus. Their functions are regulated by companion cells. So, the functions of sieve tube, conduction activity of the sieve tube is regulated by companion cell. This is because of the absence of nucleus in the sieve tubes. So, here we call sieve tubes and companion cells. Sieve tubes and companion cells as sister cells, as the functions of the sieve tube cells is regulated by companion cells. They are called as sister cells and they are ontogenetically related. Clear? Next. Yeah, if you look into the companion cells, they are specialized, and living parenchymatous cells, they are closely associated with sieve tubes. Okay, they are specialized living parenchymatous cells, they are closely associated with sieve tubes. They are connected with sieve tubes via pit fields. Between sieve tubes and companion cells, there are pit fields in common longitudinal walls. They contain nucleus and well dense cytoplasm. They maintain the pressure gradient in the sieve tube cells. Companion cells will measure the pressure gradient, will regulate the pressure gradient in the sieve tube cells. They control and support sieve tube functioning also. Now, phloem fibers are bast fibers, okay. They are composed of sclerenchymatous and dead cells, okay, and they are long unbranched cells with pointed needle like ends. Next, cell walls are thick and lignified. Xylem fibers might be, phloem fibers might be, their cell walls are thick and lignified. Generally absent in primary phloem but present in secondary phloem. Xylem fibers are present in secondary phloem. They are commercially used to make, jute, flax, hemp. Phloem fibers. Lastly, phloem parenchyma is composed of leaving elongated and tapering cylindrical cells. Cell walls are made up of cellulose and have pits. Plasmodesmata will connect two adjacent phloem parenchyma. They connect adjacent cells of intercellular transport. They store food materials like starch, resins, latex and mucilage. Phloem parenchyma is absent in most of the monocotyledons. They are present in dicotyledons, okay. Now, when it comes to types of phloem, just like xylem, primary phloem is of two types called as protophlloem and metaphlloem. Okay, they are of two types again, protophlloem and metaphlloem. In protophlloem, sieve tubes are narrow. In metaphlloem, sieve tubes are wide. So yes, this is all about tissues. In coming up class, we will know better about tissue systems and a proper comparison between dicot and monocot, stem, root and leaf. Thank you.